Hi everybody, my name is Andrew Grenfell and I'm the research mentor for the microbiome section of Biology 3004. In this video you'll hear a little bit about the kinds of experiments you'll do in the microbiome section as well as why we think those experiments are interesting. But before we get there I wanted to give you a little bit of background about me. So I was actually a CBS undergrad from 2008 to 2012. After that I went to get my PhD at the University of California, Berkeley, and then when I was done with my PhD I actually took a year to start a high throughput sequencing startup out in the Bay Area. After that, or most recently, I decided to come back to the U and work with Dr. Jeff Gralnick, as well as hopefully work with some of you in the coming semester.
First of all, let me introduce you to the Gralnick Lab. And this is our PI, Jeffrey Gralnick. And in the Gralnick Lab, we study bacteria, and more specifically, we study Shewanella onadensis. Shewanella onadensis is a gram-negative bacterium, and when it eats carbon sources, it generates electrons, just like everything else, but it has the relatively unique ability to move these electrons to outside of the cell, and this is important for many different types of biotechnological aspects, including bioremediation. For example, heavy metal contamination at polluted sites is a serious problem to the environment, and heavy metals at these sites are generally soluble, but electron transfer to these heavy metals allows them to become insoluble and more easily removed from the contaminated site. In addition, electrons generated from eating various carbon sources can also be captured by electrodes, and this allows for the potential to utilize Shewanella onadensis as an alternative energy source in the future. Up to this point, most of the studies concerning Shewanella onadensis revolve around its ability to be used in various biotechnological aspects. But what I'm interested in is, where does Shewanella reside in nature, and does electron transfer play a role in its environmental niches? And it just so happens that here at the University of Minnesota, we have one of those environmental niches readily available to us. And that niche is zebrafish. And it turns out that in and around the zebrafish, there's quite a diverse population of bacterial species. And these species include not only Shewanella species, but also a variety of Vibrio species and Aeromonas species. Both of these species have been predicted to also conduct extracellular electron transfer as Shewanella does. And it's these findings that form the foundation of the microbiome section of Biology 3004. And in our laboratory section, our specific aims are as follows. We hope to determine the composition of the zebrafish microbiome. We hope to characterize individual members of the zebrafish microbiome. We hope to characterize important microbial processes involved in zebrafish interaction, and we hope to determine how different types of compounds can influence the microbiome composition. And in this section, we envision utilizing the following laboratory techniques. And these include sample collection from both the environment and the zebrafish, and isolating bacterial species from these samples. In addition, we hope to utilize next-generation sequencing to potentially identify the microbiome composition, as well as elucidate the whole genomes of the isolated bacterial species. And finally, we envision using classical microbiology techniques, including cloning, strain construction, phenotypic analysis, and other protocols to identify the microbial processes involved in zebrafish interaction.